Hi, guys. Hi, Jay.
Hey.
Ready?
Hey, Kathy.
Looks like Kathy's at home.
I am.
She is. We're just getting back into the lab now. We just got permission to start a bunch of stuff again. So she's not doesn't have permission yet, but she'll get permission next week probably.
Jay, how are you?
Not too bad at all, to be honest. Yeah, no, things are moving along. It's springtime finally here too. So,
Good.
but yeah, not teaching this term has made the term so much better. So,
Hi, Carly. Where are you?
Hi. Oh, I'm at home.
Yeah, where's home now?
Oh, I'm still in St. John's. I didn't.
Okay, good.
Yeah. Let's go ahead and get started as well. Thank you everybody for joining us today. Our speaker is Dr. Jason Treberg from the University of Manitoba. And I will start by reading the land acknowledgement. We respectfully acknowledge that the territory and lands on which Memorial University campuses are situated are in the traditional territories of diverse Indigenous groups, and we acknowledge the histories and cultures of the Beothuk and the island of Newfoundland as the ancestral homelands of the Mi'kmaq and Beothuk. We would also like to recognize the Inuit of Nunavut and Nunatuvut and Innu of Nitassinan and their ancestors as the original people of Labrador. We strive for respectful relationships with all the peoples of this province as we search for collective healing and true reconciliation and honor this beautiful land together. We finally encourage everyone to reflect on these lands from where you are located and the Indigenous peoples for whom these lands are traditional territory. If everyone could keep their mics off for the duration of the talk, that would be appreciated, and we will wait for questions at the end. Go ahead whenever you're ready, Jason.
okay, well, thanks, Carly. And I want to thank everybody for attending as well as for the invitation. And just before I get into my presentation, I'm presenting here from Winnipeg, which is the original lands of the Anishinaabe, never get that, Oji Cree, Dakota, and Dene people, as well as the traditional homeland of the Métis Nation. So this is where we're zooming in from Winnipeg on those Treaty 1 lands, and I'll start to share my screen here, and hopefully all goes well. There we go. So I'm going to hazard a guess everyone can see that. And what I'm going to be talking about today is some of the adventures that we've been doing in trying to take some of what we would normally think of as our lab-based approaches and understandings and take it out to questions that deal with animals out in the wild. And I am a bit of an angler, and I just want to explain these photos. Anglers like myself, any catch is always a success. So this isn't sort of downplaying lab stuff, but it's really acknowledging that when we try and dip our toe into these field-based kinds of questions, it gets to be a lot harder and a greater challenge to sort of get to definitive answers. So I will just sort of give away a little bit of the ending in that we don't have on any of the stories that I'm telling you complete final answers yet. So these are all works in progress. So before I get into some data and such, I just want to talk a little bit about what I mean by metabolic indices, because I realize that may be a term that might not have intuitive information in it for everybody. And all I mean by that sort of phrase is, if you make a measurement, you'll get that data, but you can often infer things based upon the data. And so folks will sometimes refer to these as biomarkers, and they become really useful in getting broader understanding beyond the data point itself. So one that I quite like is oxygen consumption, which, just because sometimes those that know me, I can get a little fussy on language. Oxygen consumption is not a measurement of metabolic rate, but it can inform us on what the metabolic demands of the organism are. Similarly, if you've ever had any clinical chemistry done at your physician's office, they're basically using human biomarkers. They're looking at the levels of metabolites and proteins in your blood and seeing what that snapshot looks like for your physiological status. And the thing is, those kinds of data can be really powerful, but there's a lot of layers to understanding. So with the clinical chemistry, if somebody was to come back and have elevated aspartate aminotransferase in their blood, I can say, well, that might mean that there is a liver problem. But it might also mean that they're an athlete and they had a heavy training day the day before and have done some muscle damage. So even though these often have sort of what are thought of as standard interpretations, there's actually behind the scenes information that goes into those interpretations. And so what we're trying to do is find ways that we can use this kind of approach for animals out in the wild. So originally I was going to include some snippets of a bird project that we've helped on, and there's a lab working on urban and rural squirrels that I had to cut all that out just for expediency. So I'm going to just stick to some of the work we've been doing on fishes. So these are the three things that we'll look at. The first one is going to be what I would call sort of an illustration where these techniques I think work really well. The next one is going to be a little bit of a work in progress. And then this final one I'm really excited about. And it's interesting when you find results that don't always fit the pattern you expect. So for those that don't know the kind of work that goes on in my group, this kind of summarizes it fairly well. We do a lot of work on mitochondria, very interested in the trade-off between the formation of reactive oxygen species versus oxygen consumption, ATP production, things like that. So if we start with our questions, we typically start with mechanistic biochemistry questions. We come up with models of how we think things work. That usually informs our experimental manipulations. So in this case, I've just got an example of a paper where we took rodents and put them on a diet that is known to extend their lifespan. One of the things we're interested in is this sort of biology of aging. And mitochondrial function and reactive oxygen species are thought by some folks to be related to the aging process. So as we understand mitochondria better, we can do a nutritional experiment to see, well, do the mitochondria respond the way an anti-aging response will be thought of? We can then start to, as we better understand the system, start comparing different organisms. I kind of come from a comparative background, so that's really quite fun. So here we've looked at similar processes in a very, very long-lived versus short-lived mammal. And that, of course, informs us on the kinds of next mechanistic questions we've got to figure out. These, of course, start to feed back on themselves. And eventually you build this foundation of knowledge where you and the people that work in that area of science have a lot of understanding about how things work. So the next steps are actually often more straightforward. Our problem with what we're looking at with the wild animals is we don't have as much of that foundational knowledge. And this becomes a bit of a challenge because what we end up having to do is rely very heavily on other model organisms. Sometimes there'll be other fish. Sometimes we have to rely on the mammalian literature. But there are dangers about assuming that fish function and biochemically or physiologically operate the same as in, let's say, a lab mouse. So we are always kind of having to sort of carefully piece things together with the kind of work that we're trying to apply here. And again, that's kind of that foundational knowledge that probably parallels what most folks who are here see as their own approach to science. You build upon this. What I'm finding is as we try and do this wild work, we're having less of a sound foundation, but we're going to take the try anyways. And one of the reasons that I think it's really important to gamble on this, because these are a bit of a risky approach, because sometimes you'll get results that you just can't interpret clearly, are things like this. So there's a phenomenon known within fish physiology circles where dominance hierarchies can be set up when you have large amounts of fish in a tank. And these can really affect the physiology of the dominant versus subordinate animals. But what's been known for a while is if you put those animals into a far more complex environment, like where they might live normally, a lot of those differences start to disappear. And so while these dominance hierarchies are crucially important for things like aquaculture and maintaining animals in a laboratory setting properly, there's a bit of a danger of asserting that the same phenomena are taking place out in the wild. So that's why we're sort of rolling the dice and trying. How well can we sort of see what's going on out in the world? So how we approach that is basically putting a lot of reliance on reductionism. So there is a fair bit of work where people are taking physiological measurements, things like whole organism oxygen consumption rates, in the case of fish, but other sort of physiological measurements out in the wild. And I think that's phenomenal. But one of the challenges there is you're actually taking the lab out to the wild, which isn't always practical. So what we've been restricting ourselves to in what I'm talking about today is where you're not taking the lab out with you to the field. It's fun to do that, but it's not only challenging, some environments you just can't. So if we start to think along more towards isolated organs or cells, there's a lot of really interesting questions that could be addressed there, but it still needs a lot of experimental kit or gear to bring with you. You need good, reliable sources of power. It's got to be kept dry, things like that. Whereas down here, at the very, very small scale, we can take advantage of the fact that a lot of these things can be sampled, often non-lethally, which in our case is essential for some of our studies. But we can also freeze the sample and analyze it later. Now, of course, you can see we're way down at this very small scale. So interpreting what's going on and applying it back to the whole animal, that's where our challenge runs. Now, I'll just add an additional sort of complexity, which I'm assuming most would follow, but keep in mind, when we keep animals in the lab, they're under common housing conditions. So the variation also gets quite a bit smaller when they're lab housed. Out in the wild, you've got a lot more actual real biological variation in the metrics you're measuring, which makes it even harder to see clear patterns sometimes. But that's not noise, that's not bad, that's the real biological variation. So if we start to think about these small scale sort of interactions, there's sort of four main players. We've got the DNA, which is sort of the library. We've got the RNA that is made from the DNA forming proteins, and the proteins have products, often metabolites that we'll be interested in. And the metabolites can feed back affecting proteins. Proteins can affect RNA and DNA. But the thing that's really kind of crucial here to appreciate is there's very different time scales that we're dealing with here. So metabolites, some metabolites will change within seconds. Others can take days. Proteins, actual absolute levels of proteins rarely change for a specific protein faster than within hours, but usually it's more towards days. But proteins also talk to each other. So post-translational modifications, those can often be very, very fast, but they can also sometimes require more on the scale of hours to take place. RNA, we often think of RNA as a really sort of quick first response, but the reality is once RNA is made, it's got to be translated, turned into protein. So RNA levels actually don't turn over all that fast. So there are very few examples where our mRNA is actually turning over on the scale of minutes. It's usually more into the hours, and in some cases into the days. And then of course our DNA, that's more driven at the level of inheritance, generational level, or possibly mutation level, right? So we're going to focus on these two levels. And the main reason, as Bill might know, is I'm just too messy to reliably work with mRNA. So I leave that to people who are much better at working with things on a sterile bench than myself. And one of those people is Dr. Jeffrey here, who's a postdoc I co-supervise. She's been instrumental in some of the walleye work that I'll be talking about. But this is a figure that came in one of her papers that I really like because it gets at one of the problems that we run into. So she was interested in indicators of stress, and kind of emphasizing that what we want to look at when we're dealing with fish, if we want to look at stress, if we care about acute stress indicators, we can't do things like capture them in nets and then look at acute stress indicators because they're already stressed out by the time we get them. So the kinds of things that I'm going to be talking about, they have to be things that stick around and change so slowly that our method of capture hasn't necessarily influenced what we're seeing. So we would be looking for these more chronic indicators of stress or nutritional status or. So I actually won't be talking about things like lactate or glucose because, well, two reasons. There's a lot of people doing a lot of good work on that already. And on top of that, many of the situations that we're working in, we can't actually get the sample fast enough where that would be informative. So we're kind of having to go one step beyond the usually thought of indicators of condition and try and see if we can come up with indicators that are still informative, but not the typical suspects that we look to. So we'll start with what I think actually worked really well for this approach. And it actually began for me a long time ago. That's the OSC a long time ago. And that's the lab that I did my master's and PhD in. And this is actually one of the first papers that I published. It was on this compound trimethylamine oxide. And this is a small metabolite that Paul Yancey discovered a few years earlier than this. It's accumulated in deep sea animals in their muscle. And the idea is that this molecule helps proteins from denaturing. And the thing is, as you go deeper and deeper, the hydrostatic pressure causes proteins to denature. So the idea is they accumulate this stuff to counteract that. So of course, I did that work with Bill Driedzik here. Oh. Now, several years later, Paul's group did this phenomenal study on several species of elasmobranchs, captured across several different depths. What they found is these dark circles. This is the trimethylamine oxide level. And so basically, the deeper the animal, the more of this stabilizing compound there was. But the really neat thing about elasmobranchs is they also accumulate this compound urea. It's a major part of their osmoregulatory system, but it's also a protein denaturant. So the idea is shallow living elasmobranchs have a lot of this TMAO already because it is thought of as a counteracting effect on the denaturing response that proteins would have to urea. And what was really cool was Paul's group could show, as you caught these elasmobranchs deeper and deeper, that protein denaturant urea got lower and the TMAO got higher. So basically there's a balancing of, as the protein denaturing forces increase as you go deeper, the protein denaturant goes down, the stabilizer goes up. Now that was all on interspecific comparisons. So there were issues with different temperatures, different places where they were caught, all kinds of things, but nevertheless, the pattern was very robust. So going forward quite a bit, I was working with Jim Reist, who is at Fish Oceans, because he got this Beaufort Regional Environmental Assessment Project going, where they fitted this former trawler into a deep sea surveying ship. And although we were co-supervising a student looking at Arctic cod energy, we're both fans of elasmobranchs. So we said, well, let's see what happens, because they were freezing all the animals coming up for later identification. He said, well, is there anything we could do with these animals? And I said, well, it'll depend on what we get. But if they're freezing everything, let's just see what we get. And over several years, they got a few dozen chondrichthyans over the course of several summers, but they only got this one species, Arctic skate. We know there's Greenland shark up there. Jim and I were convinced we were going to see other species. That was it, just Arctic skate. But what that meant was we ended up getting individuals from a gradient. Well, ours were from about 400 meters to 1,500 meters. So what we now had were frozen samples along a depth gradient with virtually no temperature gradient, all sitting there in the freezer saying, well, what can we do with these? So we contacted Paul and said, you've got this incredible interspecific pattern. Can we look at it intraspecifically? And he said, yeah, that sounds great. Let's do it. And this is some of the data. And what we can see is as the skates get deeper, the urea goes down, TMAO goes up. And the reason I really like these particular data is urea and TMAO don't really get broken down in the muscle of these fish. So we can reasonably assert that what was there in the muscle when the fish was caught is roughly what was there in the muscle once it was euthanized on the deck. So that time frame is not a problem for these measurements. We measured a number of other metabolites which may have changed. Now that's the data on Arctic skate. When Paul sent me these data in the email, he actually said, I swear I didn't make this up. The interspecific data overlap perfectly with our intraspecific pattern, almost to the exact same regression line as you can see here. It's slightly higher urea on average. So we can show now that this pattern that was seen across species in different depths from different parts of the Pacific is the same relationship we see with these animals in the Arctic, but the same species on a depth gradient. So it becomes a really strong argument that these patterns are real depth-driven processes. So I thought that was quite fun back then. One of the questions we often got asked about after this paper was, well, how are those levels regulated? If it varies within the species, how do they manipulate that? And so, for those who haven't seen what a skate liver looks like, this is my colleague Gary Anderson dissecting one we had to dissect these while they were still basically frozen. So it was kind of a challenge to work quickly in a very cold room anyways. But they have very large livers, and the liver is where most of the urea synthesis in these animals takes place. So keeping in mind that we know that the level of, say, urea or other things in an organism's body is the balance between production, synthesis, and loss. Well, loss of urea is predominantly loss out into the ocean. We can't exactly get these fish from 1,000 meters down and put them in a box or anything. That would be the typical experiment to look for urea losses. But we could get some indicators of synthesis. So now we'll go from the metabolites over to the protein side. Now, in the comparative world, we know that as you go across different species of elasmobranchs, there are some that have very, very low urea because they're freshwater, and then the marine ones have very high amounts of urea. And we know that this basically relates to the activity of the enzymes of urea synthesis in the liver, and that's what I'm showing here. So we've got our urea and TMAO pattern there. I've just rejigged this. So this axis here, this is the amount of urea in the muscle. This is the liver enzyme activity. And what we've got, these are shallow marine species, and what they have is high activity of this particular enzyme, high amounts of urea. Down here are the freshwater species, and they're there more just as a point of reference. They won't be directly comparable on this graph for a few reasons. But then what these orangey dots are, these were the individual data by Michael Yushishin, who's an honors student in the lab. And well, I'll tell you right now, that is not a significant relationship, not a significant correlation. And even if it was, you can see it would be largely just driven by those two individuals who happen to be two fairly shallow individuals. But the problem is we didn't get a lot of critters at the high levels of urea. So I don't know if we should put too much confidence in that line. But what we can say is that by and large, this species has very low CPS3 activity, so the rate controlling enzyme of urea synthesis, compared to other marine elasmobranchs. And that coincides with their lower urea. So, and Michael did this for this enzyme as well as several other enzymes of the urea system. So it's not just unique to this one enzyme. And it was kind of a striking result of, yeah, we can actually start to say it does look like part of the reason why the urea is lower is because these animals have lower synthetic capacity. But what we didn't get is a nice clear pattern across depth. So in case you're wondering, the actual individual variation was high enough that we didn't have a strong relationship with depth. So that explains the urea pattern. What about the TMAO? What do we know about that? Well, we go back to that lab at the OSC. Well, did not want that. There's a live link buried in my talk. Hopefully that's still on point. So, yes. Back in 2006, we basically did a survey study of elasmobranch synthetic capacity. So basically, what species can make this compound TMAO? And what we actually found is there's a really strong phylogenetic linkage to who can and can't make it, with these two families having multiple representatives that do, and then all these other families, nothing. So our Arctic skates are up here in the Ragidae, so we didn't even look because based on our comparative literature, we felt comfortable saying odds are that enzyme's not even going to be in that genus. So that's an example, I think, of where this strategy kind of works. Let's get into one where it's going to be a little bit more, but trust me here. This was a really neat adventure made possible because of the work of Dr. Enders here. She's our colleague at DFO. The DFO Arctic Institute is here in Winnipeg. And Dr. Enders has a major fish tracking project in Lake Winnipeg here, as well as they have telemetry receivers all the way down the main rivers that feed into this basin. So they've been monitoring the movement of the various fishes in this region, one of which is the walleye, which is our commercially important fish. It's small scale fisheries versus what's going on out in the Atlantic, but it is of commercial as well as recreational and traditional fisheries interest here in Manitoba. Because they were capturing animals and putting in telemetry transmitters, we were able to get some blood samples from that same period. We also took some gill biopsies for some RNA type work, which Jennifer was taking the lead on that I won't talk about. So that's more kind of what Ken and Jennifer are working on. But what we were able to do is send, So the design here is we've sampled walleye through this region from north to south. I guess we started in the south and went north. So we're looking for whether or not there are differences in just the status, biological status of the animals as we move up from south to north. And what we were particularly interested in is we did a targeted metabolomic screen. So we basically got a little over 160 different metabolites quantified. And then Lillian here did some multivariate stats to start to get a sense of what we're looking at. And that's what I've got plotted here. And I'm not going to spend too long on this. So this is just a typical PCA biplot. Our first dimension here explained about 20% of the variation, dimension 2, not quite 15%. But what we found was, quite interestingly, the most northern fish separate quite well on this axis. And that's largely explained by a lot of essential amino acids. And that got us wondering, what does that mean? And this is where we run into the problem of how do you interpret this stuff? And what we realized was, if you look through the fish nutrition literature, there's not the same sort of information you would have with, say, rodents, where we can actually talk about the relative turnover rates of most of the amino acids because people have done the tracer studies. Here we don't quite have that luxury, so we can sort of infer a little bit from the mammalian studies, but then we have to superimpose the more limited fish literature. But what's really kind of interesting is if you think about what happens when a fish is not getting enough protein in its diet, there are essential amino acids which they can't make themselves, and then non-essential. What typically looks like it happens is the essential amino acids get recycled more, so they can sometimes actually look like they're building up when the fish is nutritionally limited. Now that results a little bit mixed depending on the study because these metrics are kind of fussy to do. But that got us wondering, is this telling us that there's a difference in nutritional status between these regions within the lake? So to put that into a bit of a cartoon picture, we're looking at the fish's muscle or any tissue. It's going to have protein. As that animal grows, it also has to increase its protein pool size. No protein growth, basically no growth. Protein growth equals total growth. Well, that protein is coming from free amino acids. So there are free amino acid pools in all the different cells as well as in the blood. They contribute the amino acids so the cells can make the proteins. This is all coming from the diet, so exogenous input. But proteins are always turning over, most proteins. They get damaged, they have to be recycled if they can't be repaired. So we basically have both the body protein as well as the diet protein mixing in with this free amino acid pool, which is now going to be the pool that's drawn from to make new protein. Now, if there's excess protein availability, you can also oxidize those amino acids as a fuel. And this is typically what would happen is if the fish is eating a lot, most of the protein that's coming in as diet is actually oxidized because there's only so much growth that can take place. Okay, so that's kind of amino acid and protein metabolism in a nutshell. What we wanted to start thinking about was, are there markers that we can find that would tell us about what the relative balance between these processes are? So I'm going to do basically the same kind of illustration, but a little bit more complicated, plus with some terms here. So I'm going to start with that free amino acid pool. And I'm only thinking about the essential amino acids right now. And methionine, tryptophan, and lysine, those are going to come up in a moment. So if we picture those amino acids, they are there to be making new proteins. There can be post-translational modifications of proteins. So basically stuff's added on to specific amino acids. So we often think of that as like protein phosphorylation, these switches that turn proteins on and off. But there are other modifications that take place that can be somewhat permanent. Others can be modified and removed, but we kind of have this sort of pool of modified amino acids on the proteins. And some of those modifications, the only way the body ends up clearing those is when the protein is turned over, it goes out into the free amino acid pool, and then it's broken down. Okay, so if we start to think about the oxidation pathways, I didn't want to put all the pathways down. The reason these three amino acids are kind of key is we found in our metabolomics screen a specific breakdown metabolite of each of these three essential amino acids that is a metabolite that has shown that that amino acid has gone down the pathway that has now committed it to being broken down and oxidized. So basically, if a methionine ends up producing dimethylsulfone, that methionine, the cell that methionine was being metabolized in, has decided, anthropomorphizing cells, that methionine molecule is going to be oxidized. So these will be actually markers of commitment of these amino acids to break down as a fuel. We then realized we were actually getting a number of post-translationally modified amino acids in our metabolomics screen that might inform us on whether or not the body proteins are turning over. Because again, these are modifications that take place once it's in the protein. So if they're being liberated, they're probably coming from the body protein. So these will follow the same format. These are 95% confidence intervals from our metabolomics screen. And here's our methionine, tryptophan, and lysine again. Matt here wrote this paper. We just focused in on these three sites just to keep things a little bit simpler. So this is the northernmost, an intermediate, and southernmost sample. And what we find is these essential amino acids were all higher in the northern site. But when we looked at the breakdown markers in the blood, and this is only in the blood, we get a kind of a mixed response. Not quite a clear answer of whether or not the breakdown molecules of each of these specific amino acids is changing. So You can just go methionine to dimethylsulfone, tryptophan, and so on and so forth. The panel below is the breakdown molecule. So it looks like the essential amino acids are up, but we weren't seeing evidence for higher levels of the breakdown compounds. And then once we put the post-translationally modified amino acids, we see that in this case, the northernmost group is always higher. So putting that all back into this framework, what we think is happening is these animals might be in a state where their protein turnover is actually higher than the fish in the southern basin. But it's really unclear whether or not that's higher because they're breaking the protein down for fuel, or is there simply some reason why their overall body protein is turning over more readily? And we could speculate on that, but it would be just that speculation. Now, here's the thing. I never would have set out in an a priori plan to measure these things as a test of protein turnover. But because we did the targeted metabolomic screen, we could post hoc say, well, are there things that make sense? So now we've got this nice speculation, but it's still all speculation. Fortunately, I've got a student this fall who's keen to actually do some dietary studies on fish in the lab to see whether or not when we actually manipulate things that should change protein turnover, do these specific metabolites respond in kind? So that's what I mean by this is the start of a story. This isn't the end. And for those who know the sort of partitioning of metabolism, there's a lot of reasons why this may be not necessarily what you would anticipate to work, which is why I wouldn't have planned to do this. But again, it's that fortuitous, you do the metabolite screen, and then you can start to say, well, are there hypotheses that can come from these data? Okay, so this is the last section, and it will be a little bit less dense, hopefully. So I've been doing as much sort of collaboration with folks at the Experimental Lakes region in Western Ontario as possible. And this started with some collaborations with Matt Guzzo. Basically, these are small, isolated lakes that are in about as pristine condition as you can find, that have had very, very long-term monitoring of the fish and both the biotic and abiotic parts of the ecosystem for decades and decades. So Matt was interested in knowing whether or not this is his field lab. We allowed him to have a centrifuge. That was it. Whether or not he could take blood samples and whether or not we could sort of look for stress markers or nutritional markers, whatever we could find to look to see whether or not the animals in one lake were doing better than another lake. And the reason he was interested in this is the smaller lakes, they seem to be getting this habitat crush in the middle of summer, where these are cold water fish, and the thermocline gets deeper and deeper, and the hypoxic region keeps getting shallower and shallower over the course of the summer, and these animals get this tiny little band of space where it's actually good habitat for them. And he wanted to know whether or not we could do some blood work to sort of see, is that telling us anything? So that's kind of how it started. But rather than dwelling a lot more on metabolism, I want to talk a little bit about aging again. So this photo is really fascinating. This was taken by Craig Purchase, I believe. One of those fish is 9 years old and one is 37. And the reason they know that with that precision is the first time any of these adult fish are caught, they take a fin spine so they can age it. Then they pit tag it. And now when they catch it again, they just rescan it. They take its length, its mass, and they mark its age. So we know those fish are 9 and 37 years old. So my colleague Mike, he contacted Craig and I one day and just said, we get these super old fish every year. We get all kinds of recaptures. Is there anything we can do that would be interesting? And I said, oh, yeah. And Craig's like, oh, yeah. So we basically hatched a plan to do a lot of things. I'm only going to talk about one. But the really cool part about all of this, beyond the long-term monitoring, is that 9-year-old fish has been mature for probably a year or two, if not 3. So the 37-year-old fish has probably been reproductively active for many, 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 many years, many chances to spawn. Yet these fish have very low extrinsic mortality. It's almost like, well, Craig summarized this so well. It's almost like these are the perfect environment to look at the biology of aging, but out in the wild, which is something that folks just don't do enough of. But on top of that, they're long-lived fish. Most of the biology of aging is done on short-lived critters for obvious logistical reasons, and it's usually done in lab-relevant species. Here we've got a wild environment and a non-model species. What are we going to find? Well, what we started with was a number of aspects, but what I was really keen to try is to look at telomere lengths. So telomeres have gotten a lot of attention recently in the biology of aging, as well as in sort of colloquial media. So what telomeres are is the end of the chromosome in eukaryotic cells has these repeating segments of DNA. And the reason they're thought to be there is every time that cell divides and you replicate your chromosomes, the lagging strand, you end up with this issue of there's a little nub of DNA that most cells don't have the enzymes to replace that bit of the primer. So you basically chop a little bit off of one strand on one side of the chromosome every time that the cell divides. And there's a specialized suite of enzymes called telomerases that are required to rebuild that cap. But it's thought in most adult vertebrates, go that far, that telomerase is not expressed in most somatic tissues. Now that's starting to look like it's challengeable, but when we started down this road, that was a fair argument. So we wanted to know, are the relative telomere lengths between these two fishes changing with age, especially when we've got animals that are decades old? But it was a little bit more complicated than that, because in the ectotherm world, it's starting to be recognized telomere length might also be useful as an indicator of environmental stressors, particularly toxins or possibly temperature stress. And that was important for our walleye work. So there were a number of reasons why I wanted to get this up and working. So what we did was Mike and Craig did most of the work out at ELA with some students that Craig recruited. I brought in Mike Gowdry into this. He's an excellent molecular biologist. I don't do that stuff very much, if at all. So Mike actually built up and validated the assay. And what this assay does is it measures the length of those repeats based on PCR versus what we do is we look for a single copy gene in the genome. So what you're looking at is how many copies do you capture of the telomere repeats by PCR versus a single copy gene. And you can normalize to that single copy gene and then compare one animal to another. So it's not the gold standard for those who are familiar with telomere length measurements, but it's a fairly reasonable means of comparing across, at least within the species. And so the big reveal, here we've got fish that are going on almost 40 years old. Now, if these behave like we would anticipate a good rapidly aging mammal, we'd expect a strong decline. What did we find? No pattern whatsoever. And just for those that aren't aware, fish red blood cells are nucleated. Mammals are actually the oddball. Mammals that are mature red blood cells are not nucleated. Virtually all of the vertebrates they are. So what we find is there is no pattern there with relative telomere length based on chronological age. You can see we've got this cluster of younger animals and then this cluster of much older animals. And that was just it. We could literally scan the fish lakeside and say, that's an old one. Take it for telomere length. Craig was also looking at a number of other things, including some sperm performance. And I won't go beyond that because the manuscript was literally submitted yesterday, so we don't want to mess with any of that mojo. But I can also add from the work that we've been doing with Matt Guzzo, looking across lakes, we've found that protein oxidation and lipid oxidation markers, they don't seem to relate to age. Antioxidants don't really seem to relate to age. our marker enzymes for energy metabolism, they don't seem, it seems like the old and young are actually quite comparable. So that was really quite surprising, because if you look at the standard expectations in your typical aging models, as senescence kicks in, cells just don't work as well. These fish seem like they might actually be aging very, very differently than the sort of canonical expectation. And again, trying to keep fish alive for 40 years in the lab, I don't think I would be capable of that. So with that, I'd like to thank some of these folks that weren't specifically necessarily named or photos shown. Obviously, the folks in the lab currently who are continuing on some of these projects. My funders, of course, none of this would be possible without the funding. And I just want to take another moment to just emphasize how this all required so much collaboration with folks like from Fisheries and Oceans. I would never have been able to sort of do any of this work if it wasn't for the really collaborative nature of these projects. So I think that that's really something that I can't thank them enough for opening up the opportunity. And with that, thank you for your attention, and I'd be happy to take any questions or comments.
Thank you so much. If anyone has a question, you can go ahead and unmute yourself, or you can type your question in the chat box.
Chat box.
I have a question, Jason. Based on the last part of your presentation on the longevity research, how do you think about looking for SNPs? Because I know that there is a study in people, actually in humans in Italy, that they have difference in longevity compared to another region of Italy at the same age. So they were looking for the telomeric region as well, as you do, but they found out that there is a lossy that is regulated for a specific gene, and they have a SNP is in that transcription transcriptor regulator. So that is the meaning of why they have more longevity. But would be interesting if you can look for an homologous of that gene in fish. Probably there is.
I think that's a fantastic point, and that's certainly a bit further beyond than where we are right now. But if this pattern continues, that's absolutely the sort of thing that those who get more into that type of work, yeah, that would be a great next step, because especially at the ELA, because if what I didn't mention is different lakes seem to be shifting with climate change where their maximum lifespans are changing. And that may actually show up as a form of selection where particular SNPs like that might actually really show very nicely. So it's a great point, but not something that we've started yet.
Jay, a great talk. I wouldn't have expected any different from you. You covered a lot of ground, and I've got a couple of questions for you. In your lake trout studies, have you, or are you considering using the measurements of protein turnover? Because wouldn't you expect perhaps the older animals to have slower protein turnover than the younger ones?
So unfortunately, for the samples of the really old fish, we were really focused on that project with Craig. So the ones that we do have aren't necessarily the super old fish. They're actually stuff that's been sitting in the freezer for many years. But Nahid has actually been going back and running those to see if we can see any pattern either across lakes where the life history seems to be shifting, or if we've got any patterns with age. But that's still a work in progress, because she's still sort of fine-tuning the LC assay for some of these metabolites.
Right. How do you capture a big lake trout?
That's the great part. Out at the ELA, they aren't big.
Oh,
That
really?
37-year-old fish was this big.
Oh, okay.
These teeny boreal lakes, they don't have any big prey. So once it gets to the size of shiners, that's it, or cannibalism. So they don't have a big enough food web to get to be the huge lake trout.
Ah, okay.
Yeah,
But
yeah.
nevertheless, how do you capture them?
Oh, so those are caught predominantly by short set gill nets. They do some trap nets as well. For the samples that Matt got, there are some lakes where they angle catch them as well. So I think he chose to angle catch those.
Right. Well, that would be the fun part.
Well, you can see why he'd choose that.
Yeah. I had an experiment in mind many years ago that I'll certainly never get to. And this is more of a comment than a question. And that is taking the Simone approach of injecting animals with heavy phenylalanine to track protein synthesis, and then putting the animals back in the wild, but putting balloon tags on them. So you put the tags on them, and 24 hours later, you get that animal popping up to the surface or whatever.
Yeah.
Consider doing anything like that?
I haven't, but as you said that, I remember, because I seem to recall that was going to possibly even be done out in the gut.
Yeah, that's right. Yeah.
It's a great idea, and it's the sort of thing where for most of the lakes that are the long-term monitor at ELA, they probably wouldn't let me do that because they don't want any terminal or highly risky sampling. But at the same time, there are a few lakes where they would. And it would be perfect because the reality is these lakes are so small that balloon tag, you'd be able to find it. You just need a pair of binoculars and you'd go get it. So it would be almost perfect for it. And I've actually bent Simone's ear on this turnover stuff. And yeah, we'll probably be doing some sending samples off to him just out of expediency rather than getting the assay up and running here. But
well, I'm not sure how expedient it's going to be, but okay, thank you. Great to see you.
Hi, Jay. Nice presentation. Thank you for the presentation. I was very interested in the metabolomics that you were doing. So presumably to get that many metabolites, it must be, is it shotgun metabolomics? Is it using electrospray ionization?
There's an NMR assay they're doing, and then there is I think it's a direct injection ionization on an LC. So right. it's two run, and what's kind of been handy is there's about, I think it's about a dozen metabolites that overlap between the two, because we're not doing that, we're farming it out. But yeah, the correlations between the two is amazing. So I'm actually kind of glad we had the grant to farm it out. But,
yeah, no, it's a nice, it looks like you got a nice suite. I did notice you kind of focused on, for obvious reasons, sort of on the amino acid side of the PCA. On the other side, you actually had a couple of fatty acid metabolites, the hydroxy 16, the hydroxy 14, which suggests that there's the fatty acid metabolism is in a different part of the system, and it could be quite interesting to follow that as well.
That was a great observation, and actually I don't know if it showed up in that figure, but on that dimension with where the lipids seem to be shifting, there's a couple of choline metabolites that shift too in the same way. So Lily's actually, that's going to be part of her PhD thesis, and she's currently doing her candidacy exam right now, but we're going to jump right back to the metabolomics is the hope once that's done. Yeah.
Very nice.
Hey, Kurt here, Jay. I just have a question about going back to the populations of walleye. So you got these differences in the essential amino acids. Do you know what in the environment that was related to? Is it, I mean, you said you sampled south to north. So what was the time frame between, I guess, sampling that number of areas? Is it to do with forage, or what do you think it is?
To be honest, I don't know. And this is one of the frustrations of how the DFO sampling has to work, is that basically they chase just behind ice off to catch the animals in their spawning or approaching spawning, which means that they can't get up to the north at the same time. So there's a temporal separation that we just have to live with. The nice part is, because we're collaborating with a tracking study, we'll be able to know whether or not we're seeing different populations, whether or not fish are moving.
Okay.
But that is a concern of whether or not we are seeing a regional or a temporal or both separation. I worry about it. Some others don't seem to worry as much about it because the water temperatures and things are all the same. But I don't know. They're about 3 weeks apart, if I recall.
Okay. What time of year was that, though?
In the spring.
Spring. Okay.
Yeah. So I would like to think that, well, the speculation that we did in Matt's paper was that that time of year, the northern basin fish may be doing more swimming around to find forage than the more eutrophic southern basin. And actual locomotor activity is one of the things that can stimulate turnover without net growth. So that's what we put in as a possible, but it's speculation. That's why we've got to actually do some of this in lab, as much as I've argued that out in the wild matters. No other questions? I have other pictures of fish if need be.
Is there anybody else? All right, well, if not, we will end the seminar. Thank you so much, Jason, for giving a wonderful seminar, and to everyone for your questions. And we will see everybody next week. Have a good rest of your day.
Thanks for the
Bye,
invite.
Jay. Bye, everyone.
Bye, everyone, and enjoy the Friday afternoon.
Oh, yeah, nice.